Video will be posted on the innovatebio.org website by next Monday afternoon. Thank you for joining us. My name is Dion Krupa, and I am the program manager with the Innovate Bio grant at Austin Community College in Austin, Texas. Innovate Bio is a national advanced technological education center focused on biotechnology. Both Innovate Bio and this series of talks are funded by the National Science Foundation. We encourage and support ideas and innovations you have for educating biotechnicians and encourage you to consider applying for ATE funding. Please feel free to reach out to us. Before we begin, I have a couple of housekeeping announcements. The two presentations will be back to back, followed by a Q&A. Please feel free to use the chat box to jot down questions. And lastly, additional information about future ATE project talks can be found on the innovatebio.org website, which we will put in the chat. And one last note, at the end of the Q&A, or probably during it, we will also put an evaluation link that we kindly ask for you to complete. Thank you. So it is with great pleasure that I introduce our first ATE talk speakers, Doctors David Fernandez and Jin Zhu. Their presentation is titled The Noble Project. This talk will describe the NOVA Biotechnology Leadership and Education Project aimed at recruiting, retaining, training, and placing a diverse population of students from the Northern Virginia area to work in the biotechnology industry. Welcome.
Okay, thank you so much. Before we get started, I have to apologize for two things. One of which is my voice. I'm recovering from the flu, and so this is actually me on a good day. And then the other thing is my camera is not working. It's deciding not to work for some reason. So you won't see my face, but that's okay. You can see my presentation. So if you can't see my screen, you can let me know, but I think I am successfully sharing my screen. And again, so that you can see the Northern Virginia Biotechnology Leadership and Education, or NOBLE project. I'm the PI, Jin Zhou is the co-PI, and then Vijaya Ganjreddy, who may or may not be able to join us today, is senior faculty on this project. All right. So first, a little bit about the NOVA Biotechnology Program, which this grant is aimed at. As you heard, recruitment, retention, and job placement, things like that. But we do have a pretty nice lab setup. If you look, at the screen in the background, it's kind of blurry. That's just, I made it full screen, but it's a pretty nice lab setup we have with um, uh, good equipment and lots of workstations for students. The program itself, we just had our website updated. So these are snippets from the new website. We have career outcomes for students or prospective students to look through, what kind of jobs, are involved in biotech, and what's the job market look like nowadays, particularly in the Northern Virginia DMV sort of area. In addition to that, we have two degrees that we offer. One is somewhere between 2 and 3 years, depending on how fast the student wants to go through it. It's an associate's, it's actually an applied associate's degree, which is 64 credits. And the main goal for that is job placement. If students have credits and they want to transfer to a 4-year university, they can do that with a biotech degree. But the AAS is really geared towards job placement, which is fine because there's plenty of companies in our area. The other degree is a biotechnology lab technician certificate, and that's about 1 year because it's 24 credits. And this is really geared towards if a student is coming in already with an associate's, sometimes a bachelor's. They might even have a master's degree in some cases, but they already have either a degree and or lots of credits under their belt, some of the prerequisite credits and gen ed kind of credits. And then this technician degree is really geared towards purely biotech related classes because they've already taken everything else that they've needed. In both cases, there's a summer in between, particularly for this lab technician. There's even a summer in between that 24 credits. And so in either degree, in either case, there's a mandatory um, internship that they would have to do at a local biotech company. And I'm definitely going to talk more about that. All right, let me just rearrange my screen a little bit. Okay, so taking a look at the past few years in terms of our program, it's not a very big program. And that's one of the reasons, we applied for this grant. We're trying to grow it. So let's say before the pandemic, right about when the pandemic was about to hit in 2019, we had 13 students in our cohort. And then I highlighted in red the years that you could ascribe to the pandemic. And, 3 out of 4 of those years were single digits. It's not one of those, as you guys know, it's not one of those programs where you can do too much of it online. There's a lot that you have to do in an actual lab to get those lab skills. And even 2023, 6 students in that cohort, that was really, really sort of a low point for the past several years. Um, I'm not going to try to take credit for this. Like, I don't think we can say that the Noble Project is the main reason, but I will say that May 23, so we're coming up on 1 year since this grant was awarded, we've been really pushing the recruitment, like really hard. And I'll talk about that in a little bit. And so we have a projected number of about 15 students, and they start in the fall. So we still have, the rest of spring and all of summer to try to recruit more students. And so, potentially even more than 15. And we'd love that. The more, the better. And so we'll just have to see how this turns out. Um, I was looking over our narrative. It's kind of nice to review what you wrote and what you said you were going to do every once in a while. And we actually have 8 objectives, but they can really sort of be boiled down into these 3 main topics. So the 8 objectives boil down to 3. Really, it's about increasing enrollment, as I said. And there's a few cohorts, a few groups of students that we're really aiming to emphasize. One would be minorities in STEM, and we'll talk more about how we're approaching this. Some dual enrollment students. We as recently as last night, actually, have had some dual enrollment not activities, events at our campuses, where parents and high school students who are looking to enroll in Nova while they're still, in high school, would attend. And so we're trying to recruit like 11th and 12th graders to start taking some biotech courses and see if they can join the program. We're looking for veterans and career changers. At community colleges, I'm sure people know there's a lot of adult learners, a lot of people who are mid-career and want to try something new. And in the DMV region, there's a particularly high population of veterans. And so that's another group we're looking to recruit. That has been a challenge, but that's mostly a logistical thing. So over the next couple of years, we're going to try to work harder on that. And then, of course, our own students. I have been a salesman, and I'm sure my partners in this grant have been salespeople, just with our own students, trying to push them. Maybe push is too strong a word. Trying to encourage them, to consider biotech, and we've actually had a little bit of success with that because students, for example, taking either my own uh, or Jin's cell bio class, if they don't know where they want to go after, they've already taken cell bio or are already currently taking it. And that means they've met a lot of the prereqs for the biotech program anyway. And so it's kind of a natural fit. So we're trying to recruit from our own students anyway. Um, the retention part, we're working on mentorship, tutoring. The mentorship so far has been sort of a group mentorship model, but we're looking to move towards an individual model for mentoring our students. Tutoring has been a mixed bag just because we already have a robust tutoring program at Nova. And so, to specifically gear it towards tutoring our own students, it's kind of under the blanket of Nova tutoring in general, but we're trying to collect data in terms of if any of our students are seeking tutoring from our department, the tutoring department. We've had some cohort meetings where we would discuss some topics, including AI and biotechnology. and also any special credentialing that our students may need, and a few other topics. We've done these virtually. I think with these cohort meetings, the issue we've had is low interest, but I think that is because the timing, it's often after the students have had their classes and then a lot of them work. So it's kind of hard to make them come to something else, even though it's virtual. However, with this coming cohort in fall, we're really going to emphasize the fact that these cohort meetings are mandatory for our students. And so that combined with hopefully a larger cohort number anyway, we're looking forward to more participation and also guest speakers. So far we haven't had any guest speakers. And so it's really just us with our topics like, hey, what do you guys want to talk about? How can we best support you? But I think we're going to entice them if we have some guest speakers, and we already have somebody in mind for the first couple of them anyway. I'm not going to I hope I'm not boring you because I'm going to actually highlight a lot of what's on this particular slide as we go. So just to kind of fast forward, let's go ahead to the next slide because some of these topics that you see here, I'm going to highlight specifically. Let's look at what we've been doing for enrollment. So a couple things really. On the left, you see a picture of a 4K video where we are filming. That's actually one of the lab technicians. She's not part of the program, but lab work. We're going to be filming testimonial videos. We have one student who's already given a testimonial about what biotech is and why it's a good time to be in the program. And we're also going to be filming at some of our biotech company partners, just so you have sort of background footage of their labs and that sort of thing. And Dr. Zoe has done an introduction, overview, narration, and so the finished product of the testimonial video is not there yet, but we're almost there. We have enough to show our classes, but the finished product is coming soon, and that'll be posted on our website so people can click on it and get an idea of what our program is about. We have swag, so we have black t-shirts and green t-shirts, just like you see here. I noticed one of my colleagues, Professor Lee, is in attendance, and he had input on the design, as well as Vijaya, my partner, as well. So the black and the green look pretty good. The bags, we actually have, instead of white, it's black. So we have a green bag and a black bag. And so these are promotional items that we give out and try to raise awareness. Speaking of raising awareness, we had a recruitment event. It was part of Nova's biology club. So they call themselves BioSoc, so Biology Society. And in February, we had a Zoom meeting. Some of them were there in person, some on Zoom, where we were their guest to talk about the biotech program, which was hopefully interesting. They asked really good questions. So a lot of them, hadn't really thought about biotech too much, I think, or weren't sure what it's all about. So they had some good questions for us. So that was an event that we were able to do. In terms of the dual enrollment students that I mentioned before, so this is an image of one of our dual enrollment events. This is the table that I set up. Nothing fancy. We have these flyers that we handed out and tote bags and stuff like that. This one was at our Annandale campus, and then last night we had two more. One was at Alexandria, which is my home campus, so I was there. And then another was at the Manassas campus, which my partners on the grant, that's their home campus. And that's also the home campus of the biotechnology program anyway. And so Jin sent me some pictures from the event last night. So it looked like there was a lot of people there. I talked to a few at our event. So, we're pushing hard there. I did mention I wanted to briefly talk about partnerships with minority in STEM organizations. So that I will tell you has been really fruitful. This is a biotechnology day we held in the fall with a group called SACNAS, which is Society for the Advancement of Chicanos and Hispanics and Native Americans in Science. So it's a mouthful. So their abbreviation is SACNAS. And it was fortuitous because not only was it really a fun event, but the reason we got connected with this organization is if you see this student right here, it looks like she's doing the peace sign. She was actually my cell bio student. And so somehow we got talking about the grant, and it turns out she's the local president of this group. And she also was my student. So we connected in that way, and it was really kind of nice. So here they're smiling probably because we fed them pizza. We did not have our t-shirts or tote bags by this point. So none of them had our swag yet, which is unfortunate, but that's the way things work out. Um, we just recently, this semester, in fact, just less than a week ago, I think it was Saturday, this past Saturday, we did another Biotechnology Day event, where we had presentation just like before, and some lab skills, experiments that we had these guys do. This one was for the Leading Ladies Initiative, and they are also focused on minorities in STEM, in particular high school girls. And they're actually active in about 65 different countries. And so this is Bo right here. And I think this is his mom. It's actually a mother and son team who started the Leading Ladies Initiative. And he showed up and the time as well, doing lab activities and learning about biotech. I'll just kind of go quickly because we're probably going to run out of time soon, but the partnership with these organizations that I mentioned has also borne fruit with respect to revision of some of our classes, which falls under retention of students. So there's another organization called Involve, and their founder and CEO is going to take an active role in redesigning one of our classes, our capstone class, our capstone seminar. And so we're killing a few birds with one stone because she's also going to be talking about professional skills. That's sort of her forte. Another organization where it's fortuitous because I have a current cell bio student who's in this group. So they're also focused in women in STEM as well. We have a Bio 165 course that has been updated recently to align more with FDA regulations. I think a lot of that work happened before the grant was really in place, but it's relevant because our advisory board president also teaches this course, and he was the one with credited with the redesign. And so we're looking to maybe update some courses in our retention efforts and maybe turn some more courses online if that helps. All right, kind of summarizing towards the end with professional skills training. This is a big deal and why I'm highlighting it now. So I attended the ATEPI conference in the fall, this past fall. This is our poster on the left. I apologize. I don't remember which talk this was. The picture on the right, I don't remember whose talk it was, but it was a really good talk. And this person who gave this presentation at the PI conference was talking about industry expectations for biotech students, for biotechnicians, really. And a lot of what you see on the list, you would expect, right? Troubleshooting, robotics, networking, digital, archiving, things like this, cell culture. A lot of this is pretty much what you would expect. But the number one skill, the very first skill highlighted in this talk was soft skills, so-called professional skills. And obviously the grant had been long written before that. And so this just kind of confirmed our thinking that these so-called professional soft skills are really sought after in the industry. So to that end, last summer, we had another former student, another former cell bio student who was an absolute rock star. This is Lori. She took cell bio with me, and she came back after she was done at Nova to present to some biotech students on professional skills. And the reason she had credibility for that is because she actually had an NIH internship and then went to Yale. So she's currently at Yale. I think she's pre-med at Yale because she wants to do, as you see her, the title of her talk, physician scientist is in it. So I think she's going to do MD, PhD, or just MD and do research either way, but she wants to do both. And so she came from Nova and she came back and talked to our students about some of the professional skills that it took to get an NIH internship first and then to get accepted into Yale pre-med. So I thought that was pretty cool. And then finally, to sort of finish up highlighting our connection with local biotech companies to retain students. These are some of the big things we're doing, some of the bigger things on the project. We're going to be organizing some field trips to local biotech companies. Thermo Fisher, we just got in touch with Thermo Fisher, and it's looking like that'll be a done deal in terms of at least a field trip, if not more, connection with them, including internships. So it's really the internships that I think we're highlighting We do have budgeted money to pay for these student internships that they're doing in the summer. And so I think that's going to be a big advance for our students to really get a couple things in one shot, lab skills, real life technician skills, professional skills, and then also networking. These are some of the companies we have relationships with for internships, and then hopefully maybe also field trips and maybe more. So you can see Thermo Fisher's on there. Quest Diagnostics is a big one for us, as well as Capra Biosciences in the middle. So we're really kind of building our partnerships. Okay, and then the last sorts of things have to do with preparing our students for the workforce. For this spring, if not summer, we're going to try to get a lot of our board of supervisors who are industry leaders we talked about this at our last supervisor meeting, but we're going to try to get them to come to campus and give sort of a one-day seminar for our students, talking about exactly what you saw before, which is industry needs and expectations, as well as highlighting some of what's available for students in terms of job outlook, and then to get to know our students in a networking capacity so, they can form connections. We've already been working along those lines. The picture on the right, the audience in this classroom are some of the Board of Supervisors members who took time out to come listen to student presentations. This was part of the capstone seminar where the student project was to present sort of a business pitch, a project that, hypothetically, hey, could you fund this? So sort of a business pitch to our Board of Supervisors who gave feedback on that. And then that's where we are. So since last May, we've been doing the best we can with biting off more than we can chew. So I think we were going to have the moderator talk first before we did questions, if I understood correctly. But otherwise, you can certainly feel free to put questions in the chat box. Thanks for your attention.
Thanks, David. It sounds like you're making a lot of progress.
Thank you. I hope so.
Yeah, no, it's neat. And
Great.
now it's my turn. I'll talk and then we'll have questions after that. Okay, so share my screen and hopefully I can get my slides to work without turning everything off. Okay, are you all seeing the first slide? Oh, wonderful, thank you. So there are a lot of people on the project. I'm sort of coordinating a lot of it, but a lot of the work is actually done by other people. One of whom is, two of whom are on the call right now. Dr. Todd Smith and Dr. Erica Lannon from Prairie State Community College. So we also have Margaret Bryans, Feather Ives, Erin Kamajaya, Sheila Vemu, Alyssa Dillman, and Kandaya Mann. And we have some
sorry, but we see all of your slides. We don't see just.
Oh, thank you. Thank you. I'll fix that. All right, thanks. We'll try again. Okay. Thanks for letting me know, Dion. All right, so. Okay, we'll try. How about now?
Yep.
Yeah.
One slide?
Yes.
Yep.
Thank you.
You may want to increase it, but we can see it. We see your other slides on the other side.
Oh, you do?
Yeah.
Oh, that's odd.
We can still see.
Are you in edit mode?
I didn't think so. Guess I should have checked this first. Let's see if I get out of sharing here. All right. I'll stop sharing and then go to play mode. Okay, now we'll see. All right, how about now?
Yes.
Okay, thank you. And thanks for your patience. Really, I've never done this before. I have never used Zoom, and I've never used any kind of slide presentation stuff. No, anyway, so we'll start again. So I'm Sandra Porter, and I'm going to talk about our project with developing course-based undergraduate research experiences for antibody engineering. So we have a big team. We have myself, Todd Smith, Maggie Bryans, Feather Ives, Erin Kamajaya, Sheila Vemu, Alyssa Dillman, Kendaya Mann, and we have a few other hackathon team leaders, Erica Lannon and Thomas Honorato, Danielle Kainer, and Melanie Stegman, and I'll talk about the projects that they're leading. So a question I get a lot is, what is a hackathon? And what exactly are we trying to accomplish? Which I'll get to that in a minute. I'll talk about why you might want to be part of this or have your students be part of it. Why antibodies? And what they are. And what do we mean by antibody engineering? And then I'll talk about the projects. So what is a hackathon? A hackathon is an event where teams work together to make a product in a short amount of time. This was kind of started in the software industry, where people would have like one day where they would all try to code some kind of project, and maybe it would be useful, maybe not. But it's expanded. Since then, it's expanded to a lot of other areas, especially business, education, and art, and certainly science. The NCBI has done a lot with organizing hackathons and codathons and other kinds of groups that work on collaborative science and data science projects and databases. Do a lot of hackathons as well. But I want to clarify, they're not necessarily coding projects. So some people have this misconception that it's all about coding, and it's not. We have one project that has coding in it, and the others do not. So I said hackathons are all about making a project, right? Some kind of product. What are ours? Well, ours, we want to make research projects and supporting materials to make those research projects work. And they're all related somehow to antibody engineering. They've been used in course-based research, which was the title of our grant, but there also a lot are being used in independent student research. So as a faculty member, kind of what you can do here is you can think, oh, I have this idea. I think it would be a great research project, but how do I make this work? And in the hackathon, you get to talk to other faculty about how you might do that and figure out the instructions, the protocols, what data sets you might want to use, and give it a try. So what have we learned? We have a paper that we published last summer from our first two hackathons, and people that participated, 30 some people answered these surveys. They said the best parts were the learning and the collaboration, networking, information they got, topics, they had fun. And what did they learn? Well, they learned how to use, a lot of them learned how to use a molecular modeling program, which is free from the NCBI, called ICN3D. A lot are learning, as you might expect, about antibodies and immunology and antibody engineering, about epitopes, software databases, lab techniques. And interestingly, we expected this soft skill with teamwork and collaboration skills might be a high point with students, but actually faculty were listing this as one of the most important things they thought they learned as well, which was kind of neat. Okay, so why do you want to participate? Well, you get this chance to work on developing research projects. You get to talk with other faculty who are doing the same thing, and you get students participating who will test it and do a lot of the work sometimes. You get to make new connections, learn new skills, and like I said, it is fun. So now, why antibodies? Why would we do this? Well, so since the first antibody drug was approved, there's been 164 different antibody drugs approved. And antibodies are used in 3 important areas, not just drugs, but also diagnostics and research. And this graph that I'm showing has the FDA approvals through 2003. The orange section covers the biological license applications, many of which are antibodies. There's insulin in there too, but for the most part, these are antibodies. And you can see the fraction of drugs that are getting approved every year that are antibodies or like antibodies it's increasing. Another fraction that is increasing, you can see we have vaccines. Vaccines are related to antibodies. We have cell therapies. And I think a large fraction of the cell therapies are using modified T cells, and those modified T cells have receptors that are chimeric. Half the receptor is made from the genes of, guess what? An antibody. So a lot of antibody drugs are being developed. And in Seattle, I can tell you about two of our local companies that develop antibody drugs that are engineered. One, Pfizer bought Seagen for $43 billion not too long ago. And then just the other day, Genmab bought Profound Bio for $1.8 billion. Profound Bio was started from people by Seagen. I didn't even know about this company until I saw that it had been purchased. So as far as an important area for products, antibodies are big. Okay, so a lot of drugs, right? But not just drugs. I think all of you are probably pretty good now at doing an antibody diagnostic tests, right? If you've ever done one of the COVID tests, that's an antibody test. We also use them for pregnancy tests and ELISAs. So when I was a technician way back when, my first job was to develop an ELISA to measure a protein, concentration of a protein I was purifying. So yeah, used in a lot of places. And not just drugs and diagnostics, but research. And we'll hear a little bit about that in one of our projects. But yeah, everybody uses antibodies in research, just about. Flow cytometry, imaging, microscopy, purification. It's everywhere. And of course, being ATE and focused on workforce issues, jobs. Jobs are very important. And there are a lot of jobs for people who can work with antibodies. This is a screenshot from biotechcareers.org. And I know it's a little bit fuzzy, but there's about 400 different employers in the United States in 600 locations that say they do some work with antibodies. There's more than that, but these are the ones where it's a big part of their jobs. And going to the job board that we have on the site, you can see here, this was just a random day. I could find 79 jobs related to antibodies without even really trying very hard. And the things they work on: immunology, COVID, therapeutics, cancer, all things you might expect. Okay, so what is an antibody? I know a lot of you know what antibodies are, but not everybody that comes to our hackathons necessarily does. So this is a molecular structure model of an antibody, and it's got two different chains, 4 different chains really, but 2 heavy. Those are the green and blue ones here, gray in this picture, and light chains, which you can see here and here. These are showing up in gray. And the heavy chains are called heavy because they're twice as big as the light chains, so they're heavier. And all vertebrate animals make antibodies. So they're pretty common, used in a lot of places. We humans can make up to 10 to the 18th different forms, so a quintillion different antibodies, which is pretty astounding. And why do we do that? Because they're really important. And how do we get all these different forms? Well, we have these regions here in the V regions. V stands for variable, that are called complementarity determining regions. Those are the regions that contact the antigen or the thing that the antibody binds to. Those are made by recombining segments of multiple genes, and the recombination can be sloppy. There are lots of genes to choose from, and there are mutations that have to happen after that recombination event. It's called affinity maturation, that can change the sequence of the antibody gene and make it more powerful, make it stronger at binding. So the projects, we have 8 different projects that people will be working on. One is affordable antibody engineering. This is the one Aaron Kamajaya is leading at Los Angeles Pierce College. There is an in-person component to this. So if you're near Los Angeles Pierce College, you could sign up and go do some lab work. If you're not near, then you'll work together via Zoom. We have a bunch of different fluorescent proteins, and we're working on antibodies to those proteins and using that, trying to develop a screening tool. We are working for the first time this year, also another in-person or partially in-person project on trying to make antibodies in spirulina, a kind of algae. And that one will be led by Daniel Kainer, and it'll be at Lone Star Community College. Okay, and then we have a project working with antibodies and AI. And this one, part of the goal is to develop a way to explain what AI and machine learning are to our instructors and our novice, our biotechnician students and people that haven't used it. But to do that, we're making a large language model that can tell us, will an antibody bind a specific protein? So does this, if we have an antibody sequence, does it recognize COVID or doesn't it? That's one example. And Todd Smith will be leading this. And he worked on a similar project just a few weeks ago through the NCBI Codeathon. And we have a link to the GitHub page if you wanted to check that out. I'll put that in chat later on. We're also going to have a project that Eric Lannon is leading with antibodies against pathogens. And this one we worked on a little bit already, and Uwe was part of this too, looking at antibodies against SARS-CoV-2, because there were commercial products where antibodies could block infection with SARS-CoV-2. And you can see, I'll see if this works. Here I have part of a spike protein. And here I have it binding the ACE2 receptor, which is what it would do if it's going to infect a cell. And I'll show you how an antibody can block that interaction. So here you see one of the commercial antibodies binding right where the spike protein would be binding to ACE2, and the antibody gets in the way. SARS-CoV-2 can't get into the cell because it can't bind to its receptor. But we're so done with SARS-CoV-2, just joking, actually. Right now, what we're looking at is, if I can get to the right slide. Maybe not. Oh, there we go. This year, we're going to be looking at, Erica's team is going to be looking at antibodies to H5N1, which you may have heard of. And there is a tool that we use as part of this, not just looking at the antibodies binding to the influenza proteins, but also there's a tool we use called NextStrain, where we can track viral evolution in real time. And NextStrain just got updated with sequences from the H5N1 virus isolates that were infecting cattle and the person who got infected from working with cattle. So you can go here and you can see which branch they're all coming from, which is pretty interesting. Okay, Dr. Sheila Vemu from Wabansi Community College will be working with a different way of approaching immunity, which is to look at the epitopes that the antibodies recognize. Her team will be looking at genetic factors controlling the immune response to Mpox. And her class this year has been doing a course-based research project looking at immunity to cancer. So these are really important things in developing cancer vaccines. Understanding what epitopes you might use and what populations those vaccines are going to work in. We're going to have another project on a game. This one is going to be looking at a game that was created a long time ago, but more recently is being updated, called Immune Defense, where there's white blood cells fighting pathogens. We have a project that you may have heard about a couple weeks ago on NIST CHO cells, and working on the cell line from NIST to there's all kinds of projects around that, looking at stability of the, it's a cell line that's been engineered to make this monoclonal antibody against RSV. So we're looking at stability and glycosylation, and Maggie Bryans and Lori Kelman will be working and leading this project. And then we've got Project C-Star. So this project came about because you may know you can buy it if you work in a lab and you do ordering, you can buy antibodies to a lot of different proteins, but it really helps if those proteins are from a mouse or a human. It's really difficult to buy antibodies to non-human or non-mouse proteins, like proteins that might be found in starfish or sea cucumbers. So our idea is, if we take proteins in starfish or sea cucumbers and we look for homologs, so we use BLAST to find similar proteins that have antibodies that bind to them, maybe we can modify those antibodies and make our own antibodies for research without having to purify proteins and buy mice and buy rabbits and all kinds of other things. So we're thinking about how can we engineer existing antibodies to turn them into research tools. So those are our 8 projects: affordable antibody engineering, antibodies in algae, antibodies in AI, antibodies versus pathogens, epitopes and immunity, immune defense, NISCHO, and Project C-Star. And the hackathon will be August 5th through 8th. We do expect you to be present at least 8 hours a day. Team members count on having team members around and being able to have meetings and talk to each other. Our application is up now. We like to have a good balance of students, faculty, and industry members. And usually we've had about half faculty and half students in industry. And if you have any questions, you can let me know. As long as I don't have to use Zoom or give a presentation, we'll be okay. Thanks.
Thank you, Sandy. We have about 15 minutes for a Q&A today for both of our presenters. So let me open it up. You can either raise your hand or speak up. So there was, Sandy, there was a comment by you mentioning starfish in the chat.
Oh yeah, that's a Uwe thing. Yeah, right.
What
so
kind
the
of
original,
fish is a starfish?
right? The original name was Project Starfish because that is a name people know. But Uwe convinced us that maybe we could call it Project C-Star. Although I don't know what you do with jellyfish or other kinds of things that have fish in the name, but that's okay.
Another campaign.
Right. Anyway, Project C-Star. The Spanish name for starfish is something with stars, so, and the sea, but I don't know it.
And Elizabeth, you had mentioned in the chat about soft skills training, and you shared a link with us. David's presentation mentioned incorporating soft skills with his students.
Yeah, I just thought that might be helpful. The course series Karen Leung and the group at San Francisco are putting out on Fridays could be useful to any student.
Very good. And then David, Linnea also commented that she would like to hear more about outreach activities.
Student
Okay, sure.
recruitment.
So I guess starting with our dual enrollment students, we have a pretty robust dual enrollment office at Nova. And in addition to the events where we invited people to our campuses, they sometimes go to local high schools. And that's one thing that we have not yet been able to do, but that's very much on our menu because Apparently, when working with high school students, they're not going to get that excited about career paths, but their parents will. That's one thing I learned at the PI conference last fall. And so we want to try to see if we can get into the high schools just to do some, host some kind of event where they come in the evening and we talk about our program and that sort of thing. In terms of, let's say, outreach through our own students through NOVA, at our last faculty meeting, we really emphasized for all the bio faculty, and hopefully the adjuncts would do it too, to talk about our program, look at the website. We even have a flyer that they can pass out and try to recruit from internally from our students. And that's been okay in terms of success. We've had colleagues that are sending students our way and try to see if they're interested and go through the process of interviewing them and all that sort of thing. I think with these partnerships with minority and STEM focused organizations, STEM is such a broad topic, and so they're not per se only geared towards biotech, of course, but to try to see if we can drum up excitement amongst those students who come to our campuses for our biotech days and for our relationships with their mentors, to try to see if they have any interest in biotech, and if so, to come our way as well. I will say this, though, I certainly am learning about recruitment. It is our biggest bane. I think if we can just get them in the door, there's so much we can do for them. But that's the hardest part. So I'm open to any suggestions, actually.
So are you going to track that, or you are tracking? So when students enter your program, you ask them how they found out about your program?
Right. And so that's something we can do with even though the grant was awarded almost a year ago, we're looking at this fall as our first real cohort. And so with our first so-called real cohort, that's exactly the kind of thing that we want to do, is track how they found out. And then even with the tutoring, track to see if they seek tutoring and things like that. So yeah.
We have to do the same thing, and actually
Mhm.
that's really important for grant results besides
Yeah.
program results. So that'll be really useful.
That's good.
Yeah.
Actually, the bane of your existence is actually the bane of many programs'
Yeah.
existence.
So that's what I
tutoring.
found out at this PI conference when talking to other people near my poster, presenting their own posters. It was like everybody had the same issues, and so I didn't feel so bad. But and every student is like goals now when we're recruiting. So
Great, thank you.
yeah, thank you for that, though.
And we should have a link to the survey too, maybe. Dion.
Yes, I will.
And why Dion posted that, I was going to say, remember too, even though the grant, started a year ago and you're just starting to collect the data now, you can extend your grant, so you have the free no cost extension, but then there's other things you can do to keep collecting some of this important data.
Yes, thank you.
David, I do have a question as far as you mentioned veterans, and that's typically a group that is really hard to capture unless you partner with veteran organizations within the community. And so, but even that, my experience, that's usually hard to get into unless you're a veteran yourself. Do you find that that's a group, that's a population that you've had success with?
Right. So not yet. And when I mentioned it was for logistical reasons, we have a Veterans Affairs office at NOVA, and they have, are we still recording?
Oh,
Or is the
here,
recording
I'll
done?
turn it. I can turn it off